Good day, sir, and good day, everyone. We are the Group 16, and we're going to talk about the Week 5A, which is the plant and animal organ systems and their functions, or plant and animal immune system. So, here's the specific objectives. Number one is determine the basic concept of plant and animal immune systems, and second is compare and contrast plant and animal immune systems, and last is create awareness on COVID-19 vaccines. So let's proceed to our key concept or introduction to immune system. So immune system is all living organisms are that are continuously exposed to substances that are capable of causing them harm. Most organisms protect themselves against such as substance in more than one way, with physical barriers. So for example, or with chemicals that are repel or kill invaders. Animal with backbones is gitawag siya og vertebrates and then have these types of general protective mechanism. But they also have a more advanced protective system called the immune system. So the immune system right, is a complex network of organs containing cells that recognize foreign substance in the body and destroy them. So it protects vertebrates against pathogens. So pathogens CI is infectious disease are caused by pathogens. So ang pathogens which include bacteria, fungi, protozoa, worms, viruses, and even infectious proteins called prions. And then pathogens also classes most have mechanism for entering their host and for evading immediately destruction by the host immune system. And then and then that's why the human immune system is the most complex. So although there are many potentially harmful pathogens, no pathogen can invade or attack all organisms because pathogens' ability to cause harm requires a susceptible victim, and not all organisms are susceptible to the same pathogens. So for example, kanang the virus that causes AIDS in human, so dili na siya maka-infect or maka-infect sa animals. So whereas anang dogs, cats, and mice. So similarly, humans are not susceptible to the virus that cause canine distemper or feline leukemia and the mouse pox. So there are two kinds of immunity. The first one is innate immune system and the second one is adaptive immune system. So innate immune system is the body's first line of defense against intruders. It responds in the same way to all germs and foreign substance, which is why it is sometimes referred to as the non-specific immune system. So it acts very quickly. For instance, it makes sure that bacteria that have entered the skin through a small wound are detected and destroyed on the spot within a few hours. But the innate immune system can always stop germs from spreading. So innate immune systems provides protection offered by the skin and mucous membranes, and then protection offered by immune system cells and proteins. So protection offered by the skin and mucous membrane is all outer and inner surface of the human body are a key part of innate immune system. The closed surface of the skin and mucous membranes already forms a physical barrier that stops germs from entering. On the top of that, substances like acid, enzymes, and mucus prevent bacteria and viruses from growing. So certain movements in the body also stop germs from settling. For example, movements of hair-like structures or cilia in the lungs or movement of the bowel muscles. So some fluids in the body have a similar effect, including tear fluid, so sweat and urine, which flushes the urinary systems. So protection offered by immune system cells and proteins. So if germs get past the skin or mucous membranes and enter the body, The innate immune system fights the using special immune system cells and proteins. So what happens during an inflammation? So if, for example, an area of skin becomes infected, immune system cells spring into action, or either by moving to the area or by being activated locally. So certain cells of immune system release substance to make the blood vessel wider and more leaky. So this causes the area around infection to swell because warm and turn red, visible signs of the inflammation that has developed. A fever may develop too, then the blood vessels get wider and even more arrive immune system cells arrive to fight that infection. So phagocytes is making germs harmless. Bacteria or viruses that enter the body can be stopped right away by the phagocytes, also known as scavenger cells. These special white blood cells or leukocytes, and enclose germs and digest them, making them harmless. So the remains of the germs move to the surface of phagocytes where they can be detected by the adapted immune system. So there are also other types of immune system cells that release substance to kill bacteria and other germs. But it's not only the germs that die. Tissue cells and immune system cells die and break down too. Together they remains from a yellowish fluid called pus. So the role of proteins is several proteins enzymes help the cells of innate immune systems. A total of 9 different enzymes active each other in a kind of chain reaction. One enzyme in the first stage alerts several enzymes in a second stage, each of which activates several enzymes in a third stage, and so on. This allows the immune response to grow stronger very quickly. So, the tasks of this enzyme include marking germs as targets for phagocytes, and then attracting other immune system cells from the bloodstream, now destroying bacteria cell walls to kill the bacteria, and fighting viruses by destroying the viral envelope, so the outermost layer of virus or cells that have been infected with viruses. So innate immunity consists of four types of defensive barriers, and the first one is anatomic, which is nadi ang anatomic, and then ang anatomic is skin and mucous membrane, and then sa physiologic is temperature, low pH, and chemical mediators. And then sa phagocytic or endocytic virus, and then inflammatory virus, and sa last. So, maunday na iyang mechanism. So, let's move on to the adaptive immune system. So, if ang innate gagay na is non-specific immune system or or fails to destroy the germs or dili siya general, but the adaptive is a specialized immune system that takes over. So the adaptive immune system is specifically targets the type of germs that is causing the infection. So, but to do that, it first needs to recognize the germs as such. So, this means that it's slower to respond than the innate immune system, but it's more accurate when it does respond. So, it also has the advantage of being able to remember the germs. So, the next time the adaptive immune systems face a germs it has already met, it can start fighting the germ faster. So this memory is also the reason why there are some illnesses you can only get once in your life because afterwards your body becomes immune to them. So it may take a few days for the adaptive immune system to respond the first time it comes into contact with the germ. but the next time the body can react immediately. So the second infection then usually goes to unnoticed or it at least milder. So the adapted immune system is made of T cells in the tissue between the cells, body cells, and then B cells is also in the tissue between the body cells, and then antibodies in the blood and other bodily fluids, called T lymphocytes, are made in bone marrow, and then they travel in the bloodstream to the thymus. They were mature, so the T in their name comes from thymus. So T cells have three main jobs. The first one is they use chemical messengers to active other cells of the immune system, starting the adaptive immune system response. So T helper cell na siya. And then they detect tumor cells or cells that have been infected by viruses and destroy them. So cytotoxic T cells. And then some T helper cells become memory T cells after the infection has cleared out. They remember the germ that was fought off and they are then ready to active the adaptive immune system quickly if the body is infected by the same germs again. So these cells have specific features or receptors. So on their surface that germs can attach to, like a lock that one particular key will fit. So the immune system can make a matchy T cell type for each germ within a few days of infection. And then if a germ attaches to a matchy T cell, the T cells start to multiply, making more T cells that can specifically fight the germ because only the cells that match the germ multiply. The immune response is tailor made. So if and the B cells naman as B cells is called B lymphocytes are made in the bone marrow cell. So where they matured into specialized immune system cells. They take their name from the B in the bone marrow. Like the cells, I like the T cells, there are many different types of B cells that match particular germs. B cells are activated by T helper cells. So T helper cells send signals to B cells that match the same germs as they do. This stimulates the B cells to make copies of themselves and turn into plasma cells. The plasma cells quickly make very large amounts of antibodies and release them into the blood because the T helper cells only active the B cells that match the attracting germs, so the body only makes the exact antibodies that are needed. So some of the activated B cells turn into memory cells and become part of the memory of the adaptive immune system. So the different cells of the adaptive immune system communicate either directly or through soluble chemicals messengers such as cytokinase or usually proteins, and these chemical messengers are made by various cells in body. So now, sa antibodies is antibodies protein with sugar groups attached to them. So travel around the body in the bloodstream. They are made by the immune system to fight germs and foreign substance. Antibodies can quickly recognize germs and other potentially harmful substance, and they attach to them. This makes the intruders harmless and attract other immune system cells to help. Antibodies are made by B cells. Germs and substances that can trigger the production of antibodies are called antigens. So, antibody only attaches to antigen if matches exactly, like a key in the lock of the antibody. In this way, antibodies recognize matching germs and trigger the fast response of the adaptive immune system. So antibodies have two main functions. So the first one is they make germs harmless, for example, by directly attaching the cell surface of viruses or bacteria, or by binding harmful substance made by these germs. They prevent the germs from latching onto normal body cells and affecting them. They active other immune system cells by attaching to their surface. Also, it's much easier for phagocytes to fight the germs that have a lot of antibodies attached to them. So the last is the active proteins that help in the immune system respond. So the antibodies of the adaptive immune system also help to the innate immune system to do its job. So let's move on to the lymphocytes or heart of immune system. So lymphocytes is a class of white blood cells or are the principal active components of the adaptive immune systems. So the other components are antigen presenting cells, so which trap antigens and bring them to the attention of lymphocytes so that they can mount their attack. Giunsa di ay pag recognize the lymphocytes sa antigen. So, a lymphocyte is different from all other cells in the body because it has about 100,000 identical receptors on its cellular membrane that enable it to recognize one specific antigen. So the receptors are proteins containing groups that fit into patterns formed by the atoms of the antigen molecule, somewhat like a cake fitting into a lock, so that the lymphocyte can bind to the antigen. So there are more than 10 million different types of groups in the lymphocytes of the human immune system. So when an antigen invades the body, normally only those lymphocytes with receptors that fit the contours of the particular antigen take part into the in the immune system response. So when they do, so cold daughter cells are generated that have receptors identical to those found on the original lymphocytes. So the result is a family lymphocytes called a lymphocyte clone. So with identical antigen specific receptors. So a clone continues to grow after lymphocytes first encounter an antigen so that if the same type of antigen invades the body of a second time, there will be many more lymphocytes specific for that antigen ready to meet the uh, invader. So this is crucial component of immunological memory.
Moving on to the self versus non-self, both innate and adaptive immune system responses depends on the ability of the immune system to distinguish between self and non-self molecules. Basically, the immune system has a very crucial job of protecting the body from any harmful invaders like the bacteria and viruses while leaving its own cells alone. It does this by recognizing self molecules which are part of the body and non self molecules which are foreign. Self molecules is ma-recognize siya sa mga proteins sa surface sa atong cells, which is called the major histocompatibility complex or the MHC. Kaning mga proteins is mutabang siya sa immune system ma-identify ang atong cells as friendly. While non-self, on the other hand, are seen as foreign because they are different from our own selves. Next is we will be talking about the plant immune systems. Plants have defense mechanisms to protect against disease caused by organisms like viruses, bacteria, and fungi. They produce physical barriers, antibiotics, and enzymes to fight pathogens, but their immune system is nonspecific. Plants lack antibodies and specialized cells to detect and eliminate pathogens. So that's how plants defend themselves against the disease-causing organisms like katong mga viruses, bacteria, o fungi. So, nag-employ sila og katong mga virus strategies such as the physical barriers like cell walls, production of antibiotic compounds like phytolexins and enzymes that disrupt pathogens. And unlike animals, plants don't have antibodies or any specialized cells for targeted pathogen destruction. Instead, they rely on a more generalized defense mechanism called innate immunity, which provides a nonspecific response to infections. When plants encounter microbes, some of which are harmful to their growth and reproduction, they activate their innate immune system, which has two main branches. The first branch recognizes molecules found in various types of microbes, not just pathogens, while the second branch responds specifically to the harmful features of pathogens, either directly or through their effects on the plant. Now let us move on to the next topic, which is the plant and animal immune system. Both plants and animals is has a natural defense system, which is called the innate immunity nga nagtabang sa pagprotect sa ila from the microbes nga ilang ma-encounter. So, kini siya nga defense system is makarecognize siya sa mga specific patterns nga naay nga somehow related or naay kaugnayan sa microbe. And mao ni siya ang first line defense sa microbial invasion and is well studied in vertebrates and insects. Now, recent research shows that there are similarities between how animals and plants recognize and defend against microbes. Ang mga plants is makarecognize siya sa mga certain pattern na kitaan sa microbe that are different from those plant itself. And when plants detect these patterns, they activate their defense mechanisms. And interestingly, the way plants and animals perceive these patterns and activate their immune responses share similarities, including the presence of similar receptors and additionally the molecular processes that occur after pattern detection, leading to the activation of immune response genes, are similar in both animals and plants. Now let us talk about the antigens and antibodies. Many non-self molecules comprise a class of compounds called antigens. Antigens, which are usually proteins, bind to specific receptors on immune system cells and elicit an adaptive immune response. Some adaptive immune system cells, which are B cells, respond to foreign antigens by producing antibodies. So basically, the antigens are more resilient flags that the immune system recognizes as a foreign invaders. They are often made of proteins, and when antigens are attached to specific receptors on immune cells, is matrigger nila ang adaptive immune system. And one type of immune cell, which is called the B cells, reacts to these foreign antigens by making antibodies which help to neutralize or eliminate the invaders. An antibody is a molecule that precisely binds to a specific antigen. This may target the antigen and the pathogen displaying it for destruction by other immune cells, and antigens on the surface of pathogens are how the adaptive immune system recognizes specific pathogens. Antigens T allows for the generation of responses tailored to the specific pathogen, and it is also how the adaptive immune system remembers the same pathogen in the future. So we have here a table and it shows the five major types of antibodies that are produced by B cells, which are the IgM, IgG, IgD, IgA, and IgE. Now let us start off with the IgM or the immunoglobulin M. So the IgM or the immunoglobulin M are found mainly in blood and lymph fluid. And take note that this is the first antibody the body makes when it fights a new infection. Followed by the IgG or immunoglobulin G. So it says in here that it neutralizes pathogens such as viruses and bacteria by binding to key pathogen surface proteins and preventing interaction of the pathogen with host cell. Third is the IgD, which appears to enhance the mucosal homeostasis and immune surveillance. Next up is the IgA, which mucosal response. It protects mucosal surfaces from toxins, viruses, and bacteria through either direct neutralization or prevention of binding to mucosal surface. Lastly is we have the IgE, and it is associated with hypersensitivity and allergic reactions. Also, it plays a role in immune response to parasites.
So let's move on to antibody mediated versus cell mediated immunity. Antibody mediated immunity is the branch of the acquired immune system that is mediated by B cell antibody production. The antibody production pathway begins when the B cell's antigen binding receptor recognizes and binds to antigen in its native form. There are five types of antibodies produced by B cells: IgA, IgD, IgE, IgG, and IgM. So let's focus on IgG. IgG antibodies can be further subdivided into structurally distinct subclasses with differing abilities to fix complement, act as opsonins, and etc. The major classes of antibodies have substantially different biological functions and recognize and neutralize specific pathogens. Antibodies play an important role in containing virus proliferation during the acute phase of infection. However, they are not generally capable of eliminating a virus once an infection has occurred. Once an infection is established, cell mediated immune mechanisms are most important in host defense against most intracellular pathogens. Cell mediated immunity does not involve antibodies, but rather protects an organism through the activation of antigen specific cytotoxic T cells that induce apoptosis of cells displaying foreign antigens or derived peptides on their surface, such as virus infected cells, cells with intracellular bacteria, and cancer cells displaying tumor antigens. The activation of macrophages and NK cells, enabling them to destroy intracellular pathogens, and the stimulation of cytokine, such as IFN, production that further mediates the effective immune response. Cell mediated immunity is directed primarily at microbes that survive in phagocytes, as well as those that infect non phagocytic cells. The type of immunity is most effective in eliminating virus infected cells and cancer cells, but can also participate in defending against fungi, protozoa, cancers, and intracellular bacteria. Cell mediated immunity also plays a major role in transplant rejection. Let's move on to passive versus active immunization. Passive immunization refers to the transfer of active humoral immunity in the form of ready made antibodies from one individual to another. It can occur naturally by placental transfer of maternal antibodies to the developing fetus, or it can be induced artificially by injecting a recipient with exogenous antibodies that are usually manufactured for this purpose and that are targeted to a specific pathogen or toxin. On the other hand, active immunization refers to the production of antibodies against a specific antigen or pathogen again exposure to an antigen. It can be acquired through either natural infection with a microbe or through administration of a vaccine that can consist of attenuated, weakened pathogens, inactivated organisms, or specific proteins or carbohydrates through the induced immunity. Effective active immunization often requires the use of adjuvants, which improve the ability of the immune system to respond to antigen infection. Let's move on to COVID 19 vaccines. To understand how COVID 19 vaccines work, it helps to first look at how our bodies fight illness. When germs, such as the virus that causes COVID 19, invade our bodies, they attack and multiply. This invasion, called an infection, is what causes illness. Our immune system uses several tools to fight infection. Blood containing red cells, which carry oxygens to tissues and organs, and white or immune cells, which fight infection. Different types of white blood cells fight infection in different ways.
So these are the three immune system tools.
Macrophages are white blood cells that swallow up and digest germs and dead or dying cells. The macrophages leave behind parts of the invading germs, called antigens. The body identifies antigens are dangerous and stimulates antibodies to attack them. B lymphocytes are defensive white blood cells. They produce antibodies that attack the pieces of the virus left behind by the macrophages. T lymphocytes are another type of defensive white blood cells. They attack cells in the body that already been infected. The first time a person is infected with a virus that causes COVID-19, it can take several days or weeks for the body to make and use all the germ fighting tools needed to get over the infection. After the infection, the person's immune system remembers what it learned about how to protect the body against the disease. The body keeps a few T lymphocytes called memory cells that go into action quickly if the body encounters the same virus again. When the familiar antigens are detected, B lymphocytes produce antibodies to attack them. Experts are still learning how long these memory cells protect a person against the virus that causes COVID 19. How COVID 19 vaccines work? COVID 19 vaccines helped our bodies to develop immunity to the virus that causes COVID 19 without us having to get illness. Different types of vaccines work in a different ways to offer protection, but with all types of vaccines, the body is left with a supply of memory, T lymphocytes as well as B lymphocytes, that will remember how to fight the virus in the future. It typically takes a few weeks after vaccination for the body to produce T lymphocytes and B lymphocytes. Therefore, it is possible that a person could be infected with the virus that causes COVID 19 just before or just after vaccination and then get sick because the vaccine did not have enough time to provide protection. Sometimes, after vaccination, the process of building immunity can cause symptoms such as fever. The symptoms are normally the symptoms are normal and are signs that the body is building immunity. There are types of vaccines. Currently, there are three main types of COVID 19 vaccines that are authorized and recommended or undergoing large scale. clinical trials in the United States. First one is mRNA vaccines. mRNA vaccines contain material from the virus that causes COVID-19 that gives our cells instructions for how to make a harmless protein that is unique to the virus. After our cells make copies of the protein, they destroy the genetic material from the vaccine. Our bodies recognize that the protein should not be there and building T lymphocytes and B lymphocytes that will remember how to fight the virus that causes COVID-19 if we are infected in the future. The second one is protein subunit vaccines. Protein subunit vaccines include harmless pieces, proteins of the virus that causes COVID-19 instead of the entire germ. Once vaccinated, our bodies recognize that the protein should not be there and build T lymphocytes and antibodies that will remember how to fight the virus that causes COVID-19 if we are infected in the future. Lastly, vector vaccines. Vector vaccines contain a modified version of a different virus than the one that causes COVID-19. Inside the shell of the modified virus, there is material from the virus that causes COVID-19. This is called a viral vector. Once the viral vector is inside our cells, the genetic material gives cells instruction to make a protein that is unique to the virus that causes COVID-19. Using three instructions, our cell make copies of the protein. This prompts our bodies to build T lymphocytes and B lymphocytes that will remember how to fight that virus if you are infected in the future. To be fully vaccinated, you will need two shots of some COVID-19 vaccines. Two shots. If you get a COVID-19 vaccine that requires two shots, you are considered fully vaccinated 2 weeks after your second shot. Pfizer, BioNTech, and Moderna COVID-19 vaccines required two shots. If you get a COVID-19 vaccine that requires one shot, you are considered fully vaccinated 2 weeks after your shot. Johnson & Johnson's Johnson COVID-19 vaccine only requires one shot. If it has been less than 2 weeks since your shot, or if you still need to get your second shot, you are not fully protected. Keep taking all prevention steps until you are fully vaccinated, 2 weeks after your final shot. Know your COVID-19 vaccines. So, this is a table of knowing your COVID-19 vaccine. As you can see, there are 8 vaccines existed. And so there is Pfizer BioNTech, Oxford AstraZeneca, Sinovac Coronavac, Gamaleya Sputnik V, Johnson, Bharat Biotech, Moderna, and lastly, Novavax.